Hello everyone, welcome once again to another tutorial session in Bioinformatics. In today's video, I am going to show you how you can generate phylogenetic trees using MUSCLE and PhyML. And by the end of this tutorial, you would have generated this tree that I'm showing. In order for you to follow this tutorial, you need to make sure you have a Linux PC. Please note that the tools we are going to use can also be run in Windows and macOS, but for this tutorial, I will demonstrate using a Linux environment. You also need to make sure you have MUSCLE and PhyML installed. MUSCLE will be used to generate the sequence alignment file, and PhyML will be used to generate the phylogenetic tree. You also need to have an internet browser. This will be used to access the iTOL server and render the tree to an image. You also need to have the data. This is a multi-FASTA file that contains sequences from which the tree will be generated. Now that you are set, let's begin the analysis. We will start by downloading the data. The data can be found on my GitHub page, and this is the page. I am going to leave this link in the description box so that you can use that to also download the data. We are going to download the philo6.fasta file. To download it, you will need to first click on it. And in the next page, you click on the raw button. This can be found on the right side. The next page, you will save the file. I'm using Firefox, so I'll right click and then go to Save Pages and save it on my desktop. You can also save it in an appropriate path on your PC. I have it saved. So if you should view in the graphical interface, here it is on my desktop. This file can be viewed using any text editor. Let's open one of them. These are the sequences. Now let's go to the terminal and continue from there. On the terminal, I'll first move to my home directory and I'll issue the command cd. From here, I am going to explore the data. Please note that I cd to the home directory because of where I saved my sequence. So please make sure you also adjust these commands if you are using a different location. Now let's explore the data. We'll begin by issuing the ls command. The data was saved on desktop. And so we have it here. Let's quickly look at the sequences in this file. We see grep and we indicate the paths. We have the sequences there. There are 6 of them. We use this symbol because FASTA sequences begin with this symbol. So each symbol here indicates a different sequence. If you had hundreds of sequences, then it's going to be difficult manually counting them. And therefore, we can make it very simple if we add this pipe command and count wc-l. We'll get 6, indicating we have 6 sequences. The next thing to do is to install the tools, which are MUSCLE and PhyML. If you follow the instructions for installing the tools, then when you do an ls, you'll find the apps directory there. ls apps will give us the tools. Please note that you can also use Anaconda to install these tools. There are videos also for it, and they are in the description box. Now that we have this set, we begin by performing the sequence alignment. Sequence alignment will be performed using MUSCLE. We see MUSCLE-in, and we indicate the input file, which is the philo6.faster. And we indicate -out. That's the output file name. Let's say philo6.msc.faster. We then execute. Multiple sequence alignment is now done. When we do ls, you'll find the file there. Let's clear the screen. We are now going to refine the alignment. We are going to use MUSCLE for this as well. Let me show you how. MUSCLE-in, and this time the input becomes the alignment file we generated. Then we indicate -out. We see philo6.refine.philip. Then we indicate the refine flag, indicating we are refining the alignment file. We also indicate the output format with -phy. This is the Philip format. We now execute. Refinement has been done. ls will show us the refined file. The next thing to do is to now generate the phylogenetic tree. Please note that there are tools available to do that. These tools work with different algorithms, and so the method you choose will determine the tools you want to use. Here, I am going to use the maximum likelihood method, and therefore PhyML will be an appropriate tool because it uses the maximum likelihood method for generating the trees. I'll call PhyML and I'll say PhyML-i. The input will be the refined file. I'll also indicate -d for data type, which is NT. We are dealing with nucleotide sequences, therefore we use NT. If it's proteins or amino acids, we use AA, but those ones will be done for later videos. But now we are using nucleotide sequences, so let's stick with NT. The next is to indicate the model with -m, lowercase. We use GTR. Please note there are several models in the PhyML package. and they have been designed to work with either nucleotides or amino acids, so you choose the appropriate one. GTR works with nucleotide sequences, and that is why we are using it. To know the list of models, please visit the PhyML page. I'm going to leave the link in the description box so that you can use that to read more about this particular software. The next is to indicate the optimization parameters with -O. We use tree topology T, branch length L, substitution rate R. We now run it. The trees have been generated. Let's clear the screen first. Before I even clear, notice the statement here. So it says printing the most likely tree in file. This, that. So this is the tree here. Let's clear the screen first and do an ls. We find the tree here. Please note that there's a stats file here. This contains the model parameters and other important information concerning the settings for this analysis. I mean, the generation of the phylogenetic tree. But our focus is to just use this tree here. The next thing to do is to render the tree to an image, and we will use the iTOL server for it. The tree is in my home directory, so let's quickly view it in the GUI. This is the tree. You can open it with any text editor. And this is what you will see. This can be more meaningful if you visualize it, and that is why we are going to use iTOL to do that. So let's go to the iTOL web page. On the iTOL page, we are going to upload the tree. So you come here, you click on the upload a tree button here. On the next page, you click on browse here. Then you locate your tree. Mine is in the home directory, and there's a tree. So I click on it and open. If it's successful, you have the file name displayed here. Here. Now you can upload using the upload button here. Click on it, and it will be done for you. On the right side is the control panel. You can hide it. You can show it. There is a tree. You can drag it. You can zoom in and out as well. So this server is interactive to enable you to do a lot of manipulations on your tree. We will make this quick. We want to download it, but first we want to disable this dash lines here. They are there because by default iTOL enables the use of branch length. So to make this dash disappear, we have to ignore the branch length. So we come to the control panel, right side, click on basic, and here we have branch length. Click on ignore. So we have it here. Let's pull it here. You can view in a circular mode or rectangular, which is the default, or unrooted. Let's use the rectangular. You can also download the tree as an image. So let's download it. To download, you come to export here, click on it, and in the format you select the one you want. There are several formats here. You will download the PDF, PNG, and then SVG. So let's start with the SVG. We click on it, we come to full image, click on it, and click on export. Then you save it. I'll save them with the default names. Let's download the PDF, full image, export. We save it. Let's download the PNG, full image, export. You save it. There we have it. You can also save the circular as well. Let's do that. Circular, and repeat the same steps. We start with SVG, export, and then save file. We save on desktop. Let's get a PDF. Full image, export. Save. Desktop. Let's get a PNG or bitmap here. Full image, export. Let's save it. So now let's go to the desktop to view them. So these are the files. There are 6 of them, 3 for each mode. Let's look at the PNG for the rectangular. When you open it, this is how it appears. It appears this way because it has a transparent background. So if you should put it in a document such as PowerPoint or Word document, it will be very clear. And this is an example. Notice that it's not clear. Okay. Let's open the SVG. SVG is vector graphics. Okay, so let's do it for the rectangular. You can use tools such as Inkscape to open it. I already have Inkscape installed, so I can open it. So there's the SVG. When you have the SVG format, it's easy to manipulate and do editing on the downloaded image. So here I have it as its way. I can even delete some text files here. Let me show you. I can delete some stuffs or even change the color of this line here. Notice the color has been changed. So SVG has a lot of advantages as well. For PDF, any PDF reader will do. So that's how it is. Let's open for the circular. Here we have it. the same background transparent because the PNG format is the PDF format here, and then the SVG is also here. We can open it with vector graphics tools. So I would say congratulations for reaching this far of this tutorial, and I want to thank you for watching, and I'll see you in the next session. Goodbye.